In the war in defence of scientific knowledge against creationists, there is hardly any more certain evidence of common ancestry than that from endogenous viruses, ERVs for short. I will give a brief summary now, but much more detail is available via the links in the video description below.
To understand why retrovirus DNA is such a big deal, let's first take a quick look at what retroviruses are. A retrovirus is a special type of virus that reproduces by inserting its genes directly into a cell's DNA. The virus genes become a seamless, permanent part of the host cell's genome. The cell treats the virus DNA as if it were its own. It reads the virus genes, using them to make new viruses, and when the cell copies its own DNA before reproducing, it also copies the virus DNA and passes it on as well.
Retroviruses, HIV is an example, replicate by integrating a copy of their genomes with the DNA of the cells of the host organisms that they infect. The host cells then read the integrated viral DNA and make more viruses. ERVs are the remains of viruses that have entered the host genomes of species by integrating with their germline DNA, the DNA in sperm or egg cells, and they have thereby become heritable. They can be passed down to offspring, just like the traits for brown eyes or red hair. When a retrovirus first enters the DNA of a sperm or egg cell, we call it endogenization, and we call the result an ERV. The non-fatal ones are usually broken. That's how they can be passed down to viable offspring. Now, the essential fact to note, but creationists ignore like the plague, is that when a retrovirus integrates with host DNA, it cannot target any particular spot or locus in the DNA. But the vast majority of ERVs present in the DNA, for example in humans and chimps, are in precisely corresponding loci. The only viable explanation for them is that they were endogenized in common ancestors. Common inheritance accounts for their common locations. Separate endogenizations cannot account for them. One desperate notion that creationists will appeal to is the idea that at least some of ERVs were not caused by retroviruses, but were designed into genomes to provide benefits. They call them VIGEs, not the virus-induced gene editing of respectable science, but variation-inducing genetic elements. The idea is that maybe some of these were designed into our genomes from the beginning to be reintegrated in different parts of our genomes, rather like an unfolding puzzle. Yes, elements of ERVs do provide benefits, but never a complete, undamaged, functional retrovirus. They would be fatal. And the bits and bobs that do something useful do not have to be in the same locations in chimps and humans. The VIGE idea is a great example of a violation of parsimony. In the words of Sir Isaac Newton, we are to admit no more causes of natural things than such as are both true and sufficient to explain their appearances. Therefore, to the same natural effects, we must, as far as possible, assign the same causes. But besides being unnecessary, VIGEs don't even provide a viable explanation at all. The real stakes in the heart of this idea are: 1, the reintegration would be performed by the retroviral enzyme integrase, which, as has been proven, cannot target specific DNA loci. However many ERVs could have been VIGEs, and however many are supposed to be reintegrations, the VIGE idea cannot possibly explain the common locality of the reintegrations in the two species. And 2, we have proof that ERVs are not designed in parts of genomes. The koala retrovirus, KORV, makes that clear. It is a retrovirus currently endogenizing in koalas with no VIGE in sight. Please see the links below for more info.